I would like to thank you all for being here, and also for to the organizers of iTrain. Today I'm going to speak about the single dish pipeline, the ALMA single dish pipeline, how to reprocess data using this pipeline. And well, I would like to say that this presentation, I'm presenting these slides, but I'm doing it on behalf also of Toshiki Saito from NEOJ in Japan, who is the subsystem scientist for the single dish pipeline. So he helped me. Well, we put together all these slides. We worked together to put these slides in this presentation. So he is also around. So if there are questions or things that are beyond my knowledge of the pipeline, I think he will help us answering questions. Okay, so then I will just start right away, and then I will say that most of the things that I will present here, they can be found in the pipeline users guide that you can download from the link that appears here in the presentation or in this slide. Particularly, you can go or check the chapters number 6 and 7 of this document, where it is described with a lot of detail how to rerun the pipeline, all the things that need to be done, etc., etc. But well, during this tutorial, we're going to go through all those steps, which is the main point of iTrain, to actually do an example, sort of like interactive example of how to do these steps. Well, and I will start with a very simple question: what is a pipeline? And I found an explanation in a presentation given by Vincent Gears, where, well, he says that the pipeline is an automated data reduction software for ALMA. And basically, it consists of 3 things, which is the heuristics in the form of Python scripts. And also, it has a part that is, it's called the contexts, that are used for the bookkeeping purposes of the data process, and then the weblog generator that showcases the outcome of each calibration and imaging task, so that they can be viewed in a browser. And also in the user's guide of the pipeline, it says that well, the pipeline is used for this automated calibration of the ALMA Interferometric and single dish data. And so what is this single dish data? Well, it's what we also know as the ALMA Total Power Array. I mean, it's data that is taken with the ALMA Total Power Array. So I will go a little bit more on what is this. So all these explanations or all these descriptions of the pipeline, I picture them in my head as something like this. Basically, you have the data that either comes from the interferometer part of ALMA or the single dish part of ALMA, and then it passes through this CASA that together with the pipeline. Here, the pipeline is depicted as all these Python classes, and the outcome is what we actually want. We want the images of the astronomical sources, and we want spectra and all kinds of things, the weblog and so on. Here, well, listed as calibration products, imaging products, weblog, and calibrated measurement sets, etc. And well, during this iTrain tutorial, or this iTrain tutorial consists basically of 2 big parts. And one is a brief introduction, where I will answer, or where I will try to answer what are single dish or total power observations and how they differ from interferometric observations, and what are the single dish or total power data products and how they differ from the interferometric products that probably many of us are more familiar with. And then there will be a second part of the tutorial that would be the training part, and there we will answer questions such as why does one need to rerun the single dish pipeline, or how to rerun it to improve the data products that are available in the archive, and how and what one can tweak in the pipeline in order to improve those data products. So then, well, I will start with the brief introduction, and I'm going to start with this very simple, or this question is: ALMA is an interferometer, is it? So of course it is, but it is a little bit more than that. So we know that, well, ALMA has this, the main array, which consists of around 50 antennas of 12 meters in diameter. And we know that they can be configured in different, or they can be distributed in different configurations, et cetera. Then there is the Atacama Compact Array, so-called ACA, and also known as the Morita Array, and it consists of 12 antennas of 7 meters in diameter. And then there is the Total Power Array, which consists of 4 antennas of 12 meters that work as a single dish, or they work in single dish mode. And so then basically ALMA is 3 telescopes in one. At least I like to think about it like that. So, but one may wonder, why do we need 3 telescopes in one? I mean, why don't we just have, like, for example, one? Well, the thing is that due to the nature of the observations that ALMA does, and the way that it works, is that, I mean, as an interferometer, we know that, for example, in the case of the array of the main array, if we observe, like in this case, this galaxy M100, we know that we are not seeing all the structures. There will be some structures missing that are so-called, they are filtered out due to the nature of the interferometric observations. And then in that, when this happens, one would need to observe with a more compact array. And in this case, that's why we have the Atacama Compact Array that will recover a little bit more of those extended components, but sometimes it is not enough, and we would need a single dish observations in order to recover all the emission from all the different scales, spatial scales on the sky. So here, well, we see that if one combines the 12-meter data with the ACA data, one recovers already more than just with the 12-meter. But if one adds the total power, then one recovers even more. And we still have information of the small scales that are given by the main array, so that we can have the best of the two worlds. We can recover a lot of extended structures, but we still see small details. That's why we need the three parts of ALMA, or the 3 telescopes that are included in ALMA. So what are the difference between the interferometric observations and the single dish observations? Well, these are some of them. They're not all of them, but some relevant ones that, for example, in the interferometer, one needs information of the phase of the signal coming. So then that's why one would need a phase, so-called phase calibrator. And also one would need information of the bandpass response of the receiver. Then one would need a bandpass calibrator. And also the data of an interferometer comes in a way, or in the form of visibilities in the Fourier space. So then one would need to transform those visibilities from the Fourier space, and also one would need to deconvolve the images. And then, well, and as I mentioned before, the interferometer would not detect all the emission from all the different scales, but it will filter out some scales. So then in most of, or in many cases, it will not detect all of the emission, but just a part of it. And finally, the beam of the observations are, for the interferometer, is the Fourier transform of a sampling of the sampling function, which is basically determined by the distribution of the antennas in the array. The more extended, the more separated the antennas are, the smaller the beam would be. For the single dish, well, one measures the antenna temperature, and then one would need to convert that into Janskis, for example, using a Janskis per Kelvin conversion factor. And also, there is no necessity of having a bandpass calibrator, and also the measurements are done by pointing the telescope at different locations on a grid, and then also one would need to observe an off position to as a reference to remove the contribution from the instrument and the atmosphere and other signals that one wants to remove from the observations. And the total power actually detects all the scales that are within the primary beam of the telescope, and that primary beam, the beam of the observations, is determined by the diameter of the antennas as opposed to the interferometer. So then, I mean, these are like some basic differences, and for example, the differences in how one does the observations with the interferometer and the single dish are shown in this plot, where this plot is just to show that for the interferometer, one has to observe all these calibrators, amplitude, bandpass, and then when one observes the science target or the target that one wants to study, you have to interleave observations of a phase calibrator in between. Whilst for the single dish, basically their observations, it's all the time on the source, and well, interleave with this off position or source and off position. In a more pictorial way, I show here, for example, one has the target, science target, the calibrators and flux bandpass calibrator that one observes one time, and then afterwards one moves from the phase calibrator to the science target, phase calibrator, science target, and so on and so forth. And in the case of the single dish, there is this technique of observing in a raster mode, where basically one covers all the source by pointing the telescopes in different locations. But once in a while, like at some point of this grid, one has to go to this off position and so on. So it's like you observe the target, and then you go to the off position and so on. So I mean, there are many fundamental differences in the way that the data is obtained. But also in the way that the data is analyzed and calibrated and reduced. And I don't expect you to read all the details here, but I just wanted to show how many more steps are necessary, for example, for the interferometric observations and for the single dish. Here is like a list of all the steps that were necessary to make images in interferometric observations, which goes up to 39. And while the single dish, basically, in this case, one would have 12 steps. So it's more straightforward to do the calibration, but still there are some peculiarities of this observation mode that is important to understand. Well, so then that was a very quick, brief introduction to the differences of what single dish observations are and their differences with the interferometric observations. But now we're going to start doing or working with a specific example of how to run the single dish pipeline on total power data. So then you have to, of course, you need a data set, and for this tutorial, we decided to work with a specific project that is not too heavy. I mean, there's not. It's easy to download and to handle in your probably in your system, so that it doesn't take too much space, and it doesn't require heavy computational resources. So then, if, for example, one can go to the ALMA Science Archive and search for this project with this project ID, and then there will be there are different options, but one can choose to go to the to display all the observations, where one can choose which ones one wants to download. In this case, as you can see here, this project has the 12-meter observations up here, and then the 7-meter observations down here, and the total power data, it's all the way down here. So one chooses, or in this case, I chose to download only the total power data. And but of course, like I mentioned before, in order to study all these sources that need total power, one will have also the interferometric part. ALMA wouldn't deliver, or the ALMA does not observe only in total power standalone mode. So if there is total power data, means that there is also interferometric data. So then when one downloads the data set, the total power data set, one will have in their computer a bunch of tar files like shown here. And then one needs to untar them. And when you untar them, you will have an untar directory with only the name of the project ID. So then if you navigate down the directory tree of this data set, you will find all these other directories, the science goal, the group, and the member part of the project. And then at the member level, you will have all these other directories, which is the calibration, the product, the QA, the raw and script, and the README file. So then the package that one downloads from the archive does not contain calibrated data. Those data need to be created, and if the data was calibrated originally during the QA2 process with the pipeline, then the data in principle can be the calibrated data can be obtained by restoring the calibration tables that come together in the package. And so this restoration of the calibration tables is done by running the script for PI script. So then how to know if the data was actually calibrated using the pipeline or not? Well, one easy way to do it is that you go to the script folder, and there you will see the scripts that come along with in the package. And if you see, for example, files that say like the pipeline manifest or this PPR request, that means that this data set was calibrated with the pipeline during the QA2 process, and then it can be restored just by running this script for PI using a CASA version that has the pipeline as well. So in this case, of course, it was calibrated with the pipeline during the QA2 process, and the idea is that we can restore the calibrated data using by running the script for PI. So then, but one has to be aware that the right CASA version or the right version of CASA is necessary to restore those calibration tables. So then what CASA version do I need to do this to restore the calibrated data? Well, if one wants to find out what version of CASA was used, for example, there are different ways of doing this, of finding out what CASA version was used during the QA2 process. But for example, one easy way that in this particular project, or for this particular project, is that one goes to the QA folder, and then there will be a PDF of the QA2 report. And then one just opens that PDF file, and then there will be the CASA version that was used. So that was easy. And then now one has to actually make sure that one has installed this CASA version in their own system. Otherwise, yes, you can request if you are in an institute, for example, request the people who are in charge of installing CASA versions to install this CASA version. And then if once you make sure that you have the CASA version that is necessary or that is required for restoring the calibration tables, then you change on a terminal, for example, you change to the script directory, and in that directory, then you type or you run CASA, and you have to run it with the pipeline option, which is in some cases it could be like CASA and then _ pipeline, or sometimes CASA Pi, depends on how you call CASA in your system. But here, I mean, I put 2 examples of how to do it. So when you run CASA with the pipeline, well, you have to make sure that indeed the CASA version that you're running is the one that you need to restore the calibration tables. In this case, as you can see, well, is the CASA 5.6.1-8. And also the other thing that you have to be aware or to check is that indeed these messages appear when you open the CASA, that you have this these tasks are loaded, which are tasks that are indicating that your pipeline is indeed being loaded in CASA, and then you would be able to run pipeline tasks. So I mean, just make sure that those messages appear in your terminal when opening CASA, and then you run this command within CASA, exec file, and then the script for PI, and then wait for several minutes. And then, well, since it's going to take a while, then you can just go and prepare yourself a coffee. And in Sweden, we would say that it's fika time. And so then, after several minutes of this, when you will see in the terminal, in the CASA terminal, you will see that it says the pipeline or CASA finished successfully, and you will have a new directory apart from the ones that you already had, the calibration products and so on. You will have a calibrated directory, and inside you will have these other folders, which are called products, raw data, and then you will have the calibrated and the working directory. So then, well, now we have the calibrated data, and so what kinds of problems the data products or this calibrated data may have? Well, one easy way to find out, and I would say that the way to find out this is, it's always a very good idea to check the weblog to find out what kind of problems the data may have. So in order to open the weblog, then you now check the QA folder, and there will be this file, which is it's a tar and zipped file that it says weblog, and then you untar it and uncompress it, and then you will have a folder that says pipeline. And then you change in your terminal, you change to that directory, to this directory pipeline, and then within CASA, you run this task h _weblog, and you still do this within CASA. In that directory, you will have some HTML files. You may be tempted just to try to open it with your browser, like for example with Firefox or with Chrome, and it, in the best of the cases, it will open, but you will not be able to actually move forward for long, because once you start navigating, you will see that you will find errors if you just try to open those HTML files just directly. So what it is recommended, and also in the documentation of CASA and the pipeline, it is the recommended way of opening the weblog is within CASA running this task. So then once you do that, you will a browser will open, and you will see something like this. And actually, there are other iTrain tutorials explaining on how to navigate the weblog, what kind of information there is, and so on. So it would be nice if you're, for example, watching this on YouTube, you can check in the playlist. There is one iTrain tutorial on how to explore the weblog. So then if you click in this part, the by task, you will see all the tasks that were run during the QA2 process. And then you will see, for example, that there are some numbers. Well, there are these bars here, which says QA score, and then some numbers. And those are the QA scores, but basically they indicate how well or the task went, if there were some problems while running that task, or some information that it is good that you check. So in this case, for example, we see that most of them are green, which means that basically everything went okay. But there are some others. For example, this has there is this blue bar, and then it says the elevation difference between on and off exceeds 3 degrees. And then there are some red ones that says no spectral lines are detected. So I mean, then one way it's of one first thing that one could do is, for example, click on this one, the blue one, to see what does it mean that the difference between on and off exceeds 3 degrees. And then one clicks there, and then other page will open, and then you scroll all the way down, and then you will see that it shows here an elevation difference versus time plots. If you click on that one, for example, you will see that you will see this plot, and this plot is interesting because basically it shows you what was the elevation of the source as a function of time, but also the elevation of the reference position, this so-called off position, where information of, for example, how the atmosphere and this whole system is behaving. Like basically, we want to observe this off position to remove all the behavior of the system and the atmosphere. So then we want those we want the measurement in the off position to be as similar. For example, the sky, we want the sky to be as similar as possible as to the on position, where is our source. But that is not always possible, because sometimes, for example, it cannot be very far, and also positions that are very close to our target source, they may also have, like for example, emission, like CO emission or line emission. So it would be contaminated. So we want an off position that does not have any kind of astrophysical or cosmic emission. And so that's why sometimes one needs to go to points in the sky that do not have the same elevation. And this is important. We will see why this is important to know, because if the elevation is too different, means that we are seeing through to columns of sky that are too different, and then or columns of the atmosphere that are too different, and then that may cause problems, as we will see in a few slides later. So I mean, so that's why here the web log was showing this notification. The difference in elevation is more than 3 degrees, and as we can see here, for example, then it shows that there is this QA threshold of 3 degrees, which actually triggers this notification that that we have to pay attention. So then what is the consequence of that? Well, when one checks, for example, the here in step number 8, one checks the baseline, which generates baseline tables and subtract spectral baselines. For example, one will see that even after the subtracting the baselines, one will still see this sort of like a wiggling or waves in the spectrum, and so ideally one would like to have this flat, but then one has this wiggles here in the baseline. So then the question is like, how can one remove this thing? So how to fix this problem? One thing that one first approach that one can try is that now we know that this data set or this project was reduced with the pipeline 5.6 point something, but we already have more recent versions of the pipeline. So then one can try to run a newer version of the pipeline that probably already had some functionalities added or some bugs fixed and so on and so forth. So then, but in order to run a new version of the pipeline or our data set, well, some preparation is necessary. So we go back to our to the bottom of our tree, directory tree, where one had this calibrated. Now we have the calibrated folder, the calibration, and so on. And then what it is necessary to do is to create another calibrated directory. And if you see, now we have an extra calibrated directory that I mean, you can name it the way you want. I will just name it, for example, calibrated 2. And within that calibrated folder, you would need to create other 3 folders that I named here: raw, working, and products. And those need to be named like that. Those are not the way you want. They need to be named raw, working, and products. And from the raw directory, you will copy, or you will need to copy, the ASDM.STM files that are originally in this folder. You will need to copy them to the working directory. All the ASDMs.STMs files that you find there, you copy them to the working directory. Also, you will need to copy some helper files. So this is not strictly necessary, but it's better if you have them. It's better that you copy them. It's the Jansky per Kelvin.CSV, which has the information of this conversion factor of Jansky's per Kelvin, and then also the flag template file. It is good to copy it, because if we need to add some flags, extra flags to our procedure, we would need to have that file in our working directory that is under the calibrated, the new calibrated folder. Other thing that you will need to copy is the it's a file that is called it has this code here, and then it ends with casa_pipescript.py. You copy it to your working directory here and rename it to only casa_pipescript.py. Basically, you just drop all this first part and then you just leave that. And once you have that, this file, the casa_pipescript.py file looks something like this. Basically, it has some information of the project and so on and so forth. And then here are all the tasks that are that the pipeline will run to get the calibrated data. As you can see, basically are all the steps that are shown in the web log. In principle, well, if you want just to rerun the whole calibration, you have to keep all those tasks. For example, you may drop the imaging part if you are not interested for the moment in checking the image to make images, but you just want to see the baselines, then probably you can just run all the way to this step and not to the other steps and so on. But well, in principle, for example, I left all the steps here, and then in the pipe script file, just to run all the steps anyways. And also one thing that you have to note is that here, the name of the ASDMs .sdms files do not have the extension. So these files that you copy here, the files that you copy from the raw to the working that are called like this, you have, once they are in the working directory, you have to remove this extension, this asdm.sdm, you have to remove it. So you have to rename them so that they are only named like this, like the way that they appear in the pipe script script. You can, for example, add other tasks, like there is this task HSD underscore ATM core, atmospheric correction, that did not exist in the CASA 5.6, but it exists in the CASA 6.4 or something like that. So then you can add new tasks that would probably help you to improve the data quality, or would correct potentially problems with the data. And so then in this case, for example, I added that new task, and then I run a newer version of CASA. And also, well, you need to install that newer version of CASA in your system. You call it the same way. I mean, you run it using the same command, CASA _pipeline, and you will see now that it's a newer CASA version. Again, you just have to check that you're actually loading all these pipeline tasks when you run it, and then execute the file that is now CASA _pipescript.py, and again wait for several minutes, and again it's fika time, and well, after several minutes, once you finish your fika and so on, you will have a new weblog. When you open this new weblog, what you will see is, for example, one thing is that you would notice is that there is now the extra step in the weblog, this atmospheric correction. And for this particular example, well, I just quickly checked that everything went okay. But when I check the baselines, there's still the problem. So it seems that this atmospheric correction extra step did not really help to remove this wiggling of the baseline in the band. I mean, of the spectrum. So then what else can we do? I mean, it seems that I mean, running a new version of the CASA, at least in that particular problem, it didn't help. What else can we do? Well, one other thing that we can do is to add some flags to the data to see if by flagging some data that is not that is let's say not very good, then we can somehow improve our results. And remember what I told you about this difference in the elevation, that it is well known actually, not only for ALMA, but for many single dish telescopes, that when one has when the elevation of the off position is too different from the elevation of the source, one will have this sort of like a wiggling spectrum or baseline in the spectrum. So so one thing, for example, what could do is to try to flag data that has for example, here the difference, as you can see, the difference is increasing as the time progresses. So then, for example, one could try, I don't know, to set some limit, like let's say I don't want data that has that for which the elevation difference is more than 2.5, or I don't want data for which the elevation difference is more than 2. In this case, we did an experiment and we see, okay, let's flag all the data that it has. We were very strict in our threshold. We said, let's flag all the data that has elevation difference more than 2 degrees. So then we saw, okay, so from around what time to what time we should flag. So then remember, I told you that you would add these flags in the flag template.txt file that you copied. And then here one can add that flag in manual mode. And then we say, oh, for all these spectral windows, the fields, and so on. And then the time range, we put here the time range that we want to flag and to remove that data. And then once you add those flags, you save your file, and you go back to CASA, and you run the CASA pipe script, and again wait for several minutes for it to to do the reprocessing and so on. Now using those flags, and again Fika time, and maybe you can this time just drop the cannelle bulle. Otherwise, it would be too much sugar and too much cannelle bulle for so many reprocessing. So once it finishes this run, you can check again your web log, and you will see, for example, now you will have some extra message here where it will be warning you, like you have flagged a lot of data, like 82% of the data. So, I mean, this is an experiment that we did. It's like, it is not necessarily what people should do, but it's an experiment just to show how to add flags and how they can actually mitigate some behaviors. So then how the baseline looks like afterwards, well, it doesn't look like perfect, but definitely it looks a little bit less wiggly. Now it's a little bit more showing, it still shows a little bit of those wiggles, but probably less. If we compare it, for example, before, this was before adding the flags, and this is after adding the flag. So we can see that yes, of course, I mean, the data that was taken with the off position too far from the on position, of course, it's adding all these wiggles. Of course, here in this particular example, we are sacrificing a lot of data, then your signal to noise will also decrease. And yeah, it's always a matter of finding a compromise and well, to find what kind of things work for your particular case. Just a summary of what you have to do is that, like I said, I mean, you create a new directory, like you name it the way you want, but within that directory, you have to have these 3 folders, raw, working, and products. You copy these files from the other folders that you have. You copy the ASDM files, you copy the flag templates and the Jansky per Kelvin file, and then you copy your CASA pipe script and rename it to your working directory under your new calibrated folder. You edit the CASA pipe script .py file. You can add, remove tasks in the way that you want, and so on, blah, blah, blah. You can add some, or you can add extra flags in your flag template .txt file. And you, once you do that, you save your files and you run it again within your CASA, and well, and so on, until you get something that is acceptable for you. And well, this is sort of like the basic idea, and this is the end of the tutorial. And I hope that this is useful or helping you improving your ALMA observations. And thank you very much.